Welcome to this explainer. Okay, so we've got a fascinating puzzle today. We are stepping into the shoes of a microscopic detective to ask, how do scientists actually identify the microscopic assassins and invisible helpers living all around us? You see, in their natural state, bacteria are practically completely clear. They're essentially invisible biological phantoms. So to crack the case, microbiologists have to rely on a brilliant array of chemical lenses that make this invisible world suddenly burst into color.
Here's the game plan. We're going to start with basic visibility techniques. Hit the heavy hitters of the Great Bacterial Divide, check out the tough survival pods, and wrap up with bacterial armor and tails. Let's get right to it.
Part 1, seeing the invisible microbes, the basics of bacterial visibility. So here's our fundamental problem, right? Without a chemical lens, you literally cannot see these bugs under a microscope. If you want a quick suspect profile, you grab a simple stain. It uses just one basic dye. Think methylene blue, crystal violet, or saffronin. Boom. Suddenly, a clear drop of liquid turns into a vibrant map, showing you the exact shape, size, and grouping of the bacteria in question. The whole process is incredibly fast. You prep a thin smear, let it air dry, and here's the crucial part. You heat fix it. You're literally baking the bacteria onto the glass so they can't escape. Give it a 1-minute stain, a quick rinse, and you're ready for the microscope.
But wait, what if applying that heat damages the exact evidence you're trying to examine? Well, then you use a negative stain. Instead of dyeing the bug itself, you use nigrosin or India ink to dye literally everything else. It acts exactly like a photographic negative, staining the background while leaving the bacterial cells perfectly clear and untouched.
The visual contrast between these two approaches is super cool. Simple staining gives you brightly colored targets, while negative staining completely flips the script, delivering these pristine, glowing silhouettes against a stark, dark void. Both give you the shape and size, just using totally opposite methods.
Moving on to part 2, the Great Bacterial Divide, sorting bacteria by their cell walls. Let's see how our toolkit helps categorize these microbes into 2 massive, distinct families.
Now, the gram stain. This isn't just about painting microbes a pretty color, It's a heavy-duty structural stress test. This is arguably the most important primary procedure in microbiology because it physically divides almost all bacteria into 2 main camps based entirely on their armor.
Here's how it works. First, you lock in a primary purple stain with iodine. But step 2 is where the real trial by fire happens. You flush the slide with alcohol, a harsh stress test that rips the dye away from anything without a thick, robust wall. Then you just apply a pink counterstain to catch all the vulnerable ones left behind.
And that stress test gives you the ultimate bacterial divide. Gram-positive bacteria, the ones with the thick, sturdy walls, they hold onto that purple dye for dear life, even through the alcohol wash. Gram-negative bacteria, though, thinner walls. They lose the purple dye and absorb that pink saffronin counterstain instead. So remember, purple means thick wall, pink means thin wall.
All right, part 3, tough walls and survival pods, identifying the extreme survivalists. The microbial world is packed with rule breakers that'll just laugh at a standard gram stain, requiring us to pull out some highly specialized lenses. Meet the heavy armor division. Acid-fast bacteria possess cell walls packed with this incredibly tough, waxy stuff called mycolic acid. It's a literal suit of biological armor that lets them completely shrug off standard dyes and decolorizers. No big deal to them. So, to pierce that waxy armor, the Ziehl-Neelsen method forces us to get aggressive. We literally heat the slide while flooding it with a powerful dye called carbolfuchsin, basically melting the colors straight into the cell wall. After that, we blast it with a harsh acid alcohol wash and follow up with a blue counterstain. The visual payoff here is honestly fantastic. Because that primary dye was melted right in, the heavily armored acid-fast microbes glow bright red. Meanwhile, everything else in the sample that couldn't resist the acid wash takes on the blue counterstain. Red equals armored, blue equals vulnerable. But wait, we also have to watch out for bacteria hiding out inside dormant escape pods. When things get rough, sun bacteria form this incredibly resilient inner seed called an endospore. To spot these, we use a specialized procedure. We literally steam a dye called malachite green deep into those tough, protective endospores. Under the microscope, it seriously looks like a sci-fi battle for survival.
After counterstaining the rest of the cell with safranin, you can clearly see these bright green survival spores locked safely inside, or even emerging from the vibrant red, active vegetative cells. It is absolute visual proof of their extreme resilience.
Okay, part 4, bacterial armor and scales, spotting structural accessories. Let's finish up our detective work by hunting for the tiny structural advantages bacteria use to navigate their environments. Take the capsule stain, for example. Think of a bacterial capsule like a slick molecular force field. It's an outer layer so protective that it actually physically repels dyes, like India ink or nigrosin. So instead of taking on color, the capsule just sits there as a completely clear, unstained halo. But because this biological force field is incredibly delicate, we have to change up our procedure. We mix the organism with India ink, but, and this is huge, we strictly rely on air drying. If you used heat here, you'd instantly incinerate the fragile capsule, destroying your evidence before you even had a chance to apply the crystal violet. On the flip side, sometimes the accessories we're hunting for are simply way too thin to see. Take flagella, those microscopic tails bacteria use to swim. To see those, we have to use a chemical called a mordant. The mordant essentially coats and bulks up those incredibly fine flagella, thickening them just enough so that when we apply our stain, our microscopes can finally catch a glimpse. Wrapping it up with part 5, the complete detective's toolkit. Reviewing the master key. Let's bring all these incredible chemical lenses together into one ultimate reference guide. So here's the master cheat sheet. Every single technique we've covered has a highly specific target and a totally distinct visual code. Whether you're using steam to uncover hidden spores or alcohol to test cell wall thickness, you're manipulating chemistry to reveal biology. From the clear halos of capsules to the bright red of acid-fast armor, you've now got the master key. Which brings us to the end of this explainer, and I want to leave you with a question. Knowing what you know now about how these stains manipulate biology to reveal shape, structure, and survival mechanisms, if an unknown, completely invisible microbe was threatening a patient, which tool from your new detective's kit would you pull out first to unmask it? Really think about it. I hope this changed the way you see the hidden world all around us. Thanks for joining me, and I'll catch you in the next one.